I'm now delighted to introduce our next speaker, Rosalind Moran, who's just come all the way from what she calls the oil rig, Virginia Tech, or London, where she was probably very recently also engaged in some very interesting work. Rosalind will sort of bridge more closely the, I believe, the link between neurophysiological data and inverting models of their causes. Correct?
Correct.
Okay, thank you, Rosalind.
Yes, thank you, Michael and Stefan, for the invitation to speak in Paradise. It's been lovely so far. What I'm going to talk about is a little bit more nuts and bolts. You're correct, I was at an SBM course recently, and this talk will touch on a type of tutorial style for dynamic causal modelling for neurophysiology. So for those of you who don't use physiological recording techniques and look at fMRI instead, the kind of basic principles are the same. So we're going to look at generative models, or how we generate brain data, and then how we go backwards and use Bayesian statistics, and we'll see some of the terminology that Chris was talking about in making inferences on real data given our models. So just to outline it, I'm going to show you the approach of DCM for neurophysiology, and then look in some detail at work related to what Michael and Peter have done over the past decades on neuropopulations, and just break down a couple of the equations so that you can understand how they arise from first principles and alternatives on how they may be derived. And then look at the Bayesian piece, so how do we quantify the goodness of models and model parameters, which again is related to how Chris was describing model comparison in behavioural settings. And then I'm going to actually touch on predictive coding with some work from Martha Garrido, who's also here, I saw her today, and from Brisbane, and she's pioneered some of the early DCM for neurophysiology, looking at auditory prediction errors in the context of mismatch and activity. So that'll be a kind of button-pushing exercise. You can see how these data go through the pipeline at the very end. Right, so what does a sensor say about a synapse? You might think this is a crazy premise, that basically what we have in human non-invasive electrophysiology is a bunch of sensors or electrodes, either on the scalp measuring changes in small changes in voltage and electrical fluctuations, or magnetic fluctuations that we might record from 64 to 256 channels. And we might be given some time series like these in this image, which is just a 2D map showing you sensors over time. And if we knew nothing about the brain, then what we may have to report are just the topologies of the different signals that we're measuring under different conditions. For example, in an auditory task, maybe we have some usual tones and then some weird tones, and we may have different brain responses. So in that case, you can't really say a lot about your data feature or about the synapse from this particular data feature. But luckily what we can do, and if we're Bayesian, is use priors. So our prior knowledge on how the brain works and all that we've learned about the brain from many, many OHBMs and a few books and some good teachers. And so the two principles that are embedded in DCM, obviously, are that of functional specialization and functional integration. So on the left, outside of resting state networks, some of us still do task-based designs, which only activate selective areas. We may have activations in visual or motor cortex or some decision-making areas, and we know that they're integrated. So they have to coordinate in some way in order to produce the output that we see when our subject is being scanned. So we know that there can be discrete sources of activity that might describe this kind of largely homogeneous pattern on the scalp. Also, we have principles of measurement. So we know for the case of magnetoencephalography and also electroencephalography, and what we just heard in response to one of the questions, and even in local field potentials, that it's the synchronized activity that we're looking at when we're recording sensor or electrode-based data. So essentially, we know in the brain that we have fluctuating electrical currents at the synapses. So these are excitatory or inhibitory postsynaptic potentials. And we also have fast fluctuations at cell bodies or action potentials. So the former in the order of hundreds of milliseconds and tens of milliseconds, and the latter in millisecond timescales. So when we're recording in these modalities on the left is typically the slower currents at synapses because of their spatial summation that we're recording. And so it's this coherence, and in particular this lovely image from the Hauser lab, which is part of the Wellcome Images, is showing you a bunch of pyramidal cells, and they have a lovely orientation tangential to the cortical surface, which allows for this synchronization over space and firing. And in MEG or EEG, there's about 50,000 simultaneous pyramidal cells give rise to measurable signal. So it's a lot. We're not talking the cortical column level, but we're at this mesoscale of that neural mass models hopes to generate data. But we know that the reason that there's EPSPs and IPSPs spatially and temporally coherent at the pyramidal cell dendrites is because there are other cells either talking to it from other regions, or local regions are sending signals in order to cause these fluctuating electrical currents. And so there's something we know about that, the origin of the current sources or sinks. And so going back to Neurophysiology 101, our primary excitatory neurotransmitter in the brain, in the cortex, is glutamate. It's excitatory, so when it binds to a receptor, we'll see a flow of sodium into the cell, depolarizing the cell, and we'll have an active sink around that synapse. In the converse manner, a local population of inhibitory cells will send gamma-aminobutyric acid, or GABA, the inhibitory neurotransmitter, and open or bind receptors opening chloride channels, which will result in a active synaptic source and distributed sinks. And we know the brain isn't quite that simple. There's a whole host of other modulators that change excitabilities of cells and indeed their synchronization, things like classical neuromodulators, acetylcholine and dopamine and noradrenaline, things that come from the brainstem that Chris just showed in the lovely work by Sandra Iglesias. And there's many other things, hormones and neuropeptides, that can contribute to the fluctuations in voltage that you might measure in a typical EEG or MEG or indeed local field potential experiment. So, and this slide is borrowed from Stefan. Very nice slide, just to compare the two approaches from DCM and the general motivation in comparison to conventional studies where you might look at latencies and peaks in data and report data features, like, for example, the size of the P1 or N1 in a visual evoked potential task. But in DCM, the idea is to see how these features arrive from connected brain sources, because we know a lot, as I've just summarized, about functional segregation, functional integration, and the biophysical properties that generate fluctuating changes in currents. So eventually, these signals have to be in some way responsible for the kind of things you measure non-invasively. So what connectivity exactly? What we're talking about is, as I mentioned, synchronized activity in connected cell assemblies that result in dynamic sinks and sources in the afferent population. So the models in DCM for EEG and MEG don't describe one synapse. That's not the purpose of the generative model because it's not realistic in terms of our spatial resolution. But we want to try and describe the kind of data that we would see from a beam-formed image or some source-localized or source-extracted time series. So what we do in DCM is to try and model the underlying dynamics so that we produce a generative picture of how those data arose using our EEG or MEG. So we'll build a generative model for spatiotemporal data and fit it to evoked response or indeed other types of data features, as I'll mention in a second. And the basic underlying assumptions are of functional segregation and integration. So we think there's a few sources in our models that are generating those temporal dynamics. We'll describe the temporal dynamics using differential equations. And then we're going to project the sources through to the sensors or the electrodes using basic physical laws. So these are typically boundary element models or shell models that describe the passage from volume currents in certain areas out to what you're measuring in your sensor space. And then the last piece is to do the inversion. So once you've gotten some data from your group of subjects, how do I go back and look at the connectivity in populations of synapses and what strength are they in different patient populations or in different conditions of experimentation. So the framework is quite general, and the talk today, as I said, will focus on the right-hand side, looking at EEG and MEG. But typically, DCM uses this generative model approach and a Bayesian inversion piece in both fMRI and EEG or MEG, where in the case of fMRI, you have quite a simple neuronal model without much of the detail that you see in the EEG or MEG. So these are fun to describe, and I'll go on to those now. But the main idea is that you're getting away from data space and forming a generative model that can generate lots of different types of data. So at the moment in DCM for neurophysiology, it can generate event-related potentials. It can also generate spectral or cross-spectral features in your data, like your favorite theta or alpha or gamma bands. And then a recent addition by Carl Friston is a DCM for time frequency modulations, which is essentially a combination of the former two, where you're looking at the time-locked and induced responses to particular stimuli. So the neurals of ensembles and their population activity come from the kind of work like Peter Robinson is famous for, and Michael Breakspear. And essentially they're based on a set of neural state equations. As I said, we use different inputs to generate different data features. So we have this underlying generative model where we can use a brief temporal pulse to generate the ERP, or we can have no exogenous input but have endogenous white and coloured noise, assuming to drive the dynamics, where we could look at, for example, sleep or different conditions that can be summarised in spectral features. And then in time frequency graphs, we can produce both time modulated and endogenous fluctuations, giving us our data features. And really, there's a suite of these models at this stage. There's also some neural field equations as well as neural mass models. And this is just to show that the flavour that I'm going to say today is only a subset of what's available for push and play in SPM. So the scale of DCMs and the neural mass models really are at the mesoscale. So we know that the neuron has many number of states and attributes like potentials and conductances, but we don't model all of them, but we take this population approach and also describe activity in a single point in the neural mass models. So we know the microscale, we've looked at the different sodium chloride currents, for example, that can generate EPSPs and IPSPs. But at a mesoscale, we recognize that these are populations of cells that are connected together and conducting different ion channels. And this is the center of connectivity in DCM. And so prior to doing this analysis of connectivity, we have to know at the macro scale where the activations are. So what are our discrete number of sources that are active under this particular task? And then the really basic underlying generative model at the mesoscale is given to us by two very simple dynamic operations. One of them is actually a static operation, which is a sigmoidal description of a population firing rate. So we have some membrane potential on an afferent or rather efferent population, which generates a firing rate. And this gets convolved in the afferent population by what we're calling a convolution operator, which describes the postsynaptic EPSP or IPSP. And in DCM, we just have a scaling factor on this EPSP or IPSP that serves as the connectivity parameter. And that's what we mean when we talk about effective connectivity in the context of DCMs for electrophysiology. And so by convolving the firing rate of the incoming population with this postsynaptic response, we can then transform the second population again through this sigmoidal function for it to produce a firing rate. So we have a couple of connected dynamics. We typically have 3 or 4 populations doing this within a region, and then large pyramidal cells sending extrinsic connections to other regions. So the point of the sigmoid, this is the population analog of the single cell heaviside function with an all or nothing action potential. And it can be derived a couple of ways from the Fokker-Planck with some assumptions, or I thought it might be nice just to mention this Wilson and Cowan paper, which basically said that the sigmoidal firing curve arrives by assuming an average potential excitation in each cell and then a distribution of individual neuronal threshold potentials. So it's not like it either fires all or nothing. When you're talking about an ensemble or a population, you can have a distribution of threshold potentials, or perhaps you have a distribution of the number of synapses per cell, and the threshold is then integrated over different excitation levels. And both of these scenarios arise with a sigmoidal description when we think that the distributions are unimodal. So this parameter is important then here, rho 1, which is describing the slope of the sigmoid and describes basically the precision or the inverse variance of these distributions. So it's essentially how synchronized are your distributions, and it can be affected by rho 1 in our model. 5 more minutes. Run. Okay. So writing out these different operators, we have a convolution on a postsynaptic response, and it's convolved by the incoming sigmoidal firing rate. And again, as I mentioned, we can do this for inhibitory cells where we have a certain time constant. This is a parameter in our model, as well as the average size of the postsynaptic potential. And these are synapses activated by GABA from inhibitory interneurons. And we can have, in contrast, synapses, for example, on this population that are activated by incoming glutamate. And so our postsynaptic potential will be positive. So together we start getting these, we can enumerate via the convolution operator first of all, a second order differential equation, which we break into 2 first orders. And what we're describing is changes in postsynaptic potentials here in the pyramidal cell layer due to inhibitory activity, excitatory neurotransmission, and then the net change that we're going to eventually see in our sensors when we transmit through our boundary element or whatever forward model we prefer. So this is just to outline one source in DCM for EEG or MEG. We have these connectivity parameters, which I said are nothing more than weights on the EPSPs and IPSPs, or the sinks and sources from the other potentials. And again, like Chris's function, these DCMs are modular, and so if you don't like how this is connected and you have a different piece of cortex that you're interested in, you can rewire these diagrams according to your own specifications. So this is the sum total of the source used in DCM for EEG or MEG. And you can see it has quite a few parameters, but we have this Bayesian piece which allows us to put priors where we know their true values. Also, we have a new addition of predictive coding-based models where we have specific forward and backward pyramidal cell populations which might be enable some specific testing of the ideas Chris mentioned in the last talk. Okay, so the full generative model basically has noise. It can be white noise or a impulse response, and then some sources connected to one another, which in turn lead on to a forward model to give us activation at the level of the scalp. And can produce, as I said, different types of data depending on the input. So if we have different models, we can then do our Bayesian inversion to try and estimate both the model parameters. So how strong are the connections between cell populations in a region, and how strong are the connections between different regions. And we can do model testing, so enumerate different models. For example, we may have 4 source models where we have different conditions modulating different connections, and we can write those down by describing different hypotheses on how these data were generated. And so you've already seen Bayes' rules, so I won't dwell on it, but just to say that we can use the wealth of prior knowledge on neurophysiology, like what's an EPSP or IPSP's presumed time constant, for example. GABA channels close a little bit faster than AMPA channels, and we can embed those into our models, and we do in DCM for EEG. The Bayesian inversion piece is prescribed by defining the likelihood, which depends on how you connect your regions together. So it'll have different parameters depending on whether your connection is existing or not. And then we have our spatial, and it's just a linear mapping through the lead field. And by specifying our priors, what we do is use a variational Bayesian method to estimate the parameters of the model and also the model evidence. And I won't go through all the equations here, just suffice it to say that we use a variational Bayes approximation for posterior parameter estimation. So that means that we separate out two different parameter sets in our DCM. One of them is the observer noise, and then there's everything else in a large multivariate Gaussian. And we do like an iterative expectation maximization on a cost function known as the free energy. And so at the end, what we're getting is the ability to do inference on models. So we can use the free energy to compare one model where there might be a certain connectivity architecture to another model where it's different. But we can also do inference on parameters. So once we've optimized our cost function, we have an approximate posterior, so we can ask if particular connections exist, if they're stronger than in some other task condition, or indeed if they're different in different subjects. I won't go into the complexity term. We can go look at that offline. But just to show you some early work that demonstrates DCM very nicely, and it won't take long, it's a mismatch negativity or MMN paradigm where participants were listening to standard tones of a certain frequency interspersed with these deviant tones occurring less frequency and less frequently and having a slightly different tonal representation. This was work by Martha Garrido, originally published in Neuroimaging, where she compared models for deviant versus standard responses. Specifically, she was interested in whether the difference between that response to a deviant tone or a standard tone happened along forward and backward or backward connections. And so this is just to show you the interface. Up here we specify our data on our model. We specify what time we want to model in our evoked responses, so we can have very slow or long responses. And we can outline a design matrix. It can be parametric or model-based, just as you can in fMRI analysis. And then once you specify where your sources are, you can connect them via this kind of nice little radio button box here. And then hit invert. And hopefully what will happen is the F rises. So you should be getting, in your expectation maximization algorithm, in the MATLAB command window, increases in your, you probably can't see these numbers, but increasing Fs. And pictorially, you'll see that your data and your model are beginning to converge. So you're getting good fits to your sensor or scout-based data here in the middle. You can also see some hidden states in the model. And then at the end, you can look at trial-specific effects. So I've just highlighted those in magenta there. And this is just interesting based on what we've seen in terms of predictive coding, that what Martha found in her data, and indeed this is downloadable online on the SPM webpage if you want to practice, that the backward connections here decreased under rare events, but in rare events, so things where you had high prediction error, you saw increases in forward connectivity and also increases in the synchronization of the two early auditory cortex populations. And finally, this model group comparison is again developed in the paper, which just shows that the forward and backward connections both change under deviant compared to standard conditions. So just to summarize DCM, it enables hypothesis testing about how brain sources communicate. You can put your own underlying neural models into the code, if you like. And these, at the moment, are based on neurobiologically plausible dynamics that arise from considering distributions across cell populations of certain key parameters like firing thresholds. And also we can look at differences between modulation and the inference then using Bayesian statistics, or it can be done indeed using classical statistics. Okay, I'll thank all my colleagues and you guys for your attention. Thank you.
Okay, thank you. Thank you very much, Rosalind. And we have a few moments before the break. I will finish in a moment, so if we could just hear from the floor, and then you could also approach some of the speakers before the break. Yes?
Hi, so very interesting stuff. I always wondered with DCM, it looks like for your forward model, there's a lot of sort of detailed assumptions, I guess, built in about, like you said, firing rates and things like that, and that vary depending on where you plop your region of interest for. So I'm just wondering how sensitive the whole endeavor is to the accuracy of your forward model, and have you sort of tweaked that and tested to see if it's robust to, different I mean, if maybe you get the firing rate wrong or something like that.
Yeah, so typically what we do in developing one of these models is to look at its what types of data ranges it can generate in certain parameter ranges. And what you see is that the data, as you want them to be, are sensitive to certain key parameters, particularly time constants. But we're fairly certain that they're representative of real neurophysiology. The kind of, it may not be exactly right on the priors we choose, but some of the orders of magnitude, like tens of milliseconds versus 100 milliseconds for others, are okay. In terms of developing the models with other basically sources to tell us what we're doing, yeah, we always go back and do anatomical checks. So the new 4-level model is informed by work by people like Henry Kennedy, who've actually done track tracing studies to see what's the proportion of inputs from deep pyramidal cells to backward regions versus forward pyramidal cells, from superficial pyramidal regions. So I think the best example of a big review of the idea of a canonical, so actually we use the same model at the moment by default in every neocortical brain region, and so this idea of a canonical microcircuit is itself kind of contentious and controversial, but we've tried to summarize everybody's idea of what happens in if there is a canonical microcircuit or one circuit to describe neocortical transmission through the layers. The Bastos paper in Neuron in 2011 put a lot of different data together, including optogenetics and stuff. Okay. And then the validations we do on top of that are changing parameters by, this is work I've done, by giving people drugs, for example. You give them galantopine, you change their cholinergic system, and you see can you recover kind of commensurate changes in parameters.
Sounds like fun. Thank you.
It is. Thank you.
Thank you. And I guess in terms of giving people drugs, there's coffee out at the break.
Excellent.
I know we're supposed to say mahala, but when I came in yesterday and asked someone where there was a good lunch, he said there's a banging pokey around the corner. So being now in Hawaii, I'd encourage you to come back in half an hour. We have 2 more banging talks by Stefan and then by Petra. So I look forward to seeing you then. Thank you.